you so much for coming today. For today's program, the Neuroscience of Fear. So this is part of the Harvard Brain Science Initiative's Neuro 101 series. And today we're really excited to have with us Dr. Kerry Ressler. Dr. Ressler is the James and Patricia Poitras Chair in Psychiatry and the Chief of Scientific Officer at McLean Hospital. He's also a professor of psychiatry at Harvard Medical School. And his work, he has a deep interest in translational research, which basically brings together the worlds of molecular neurobiology and studies conducted in animal models with the human genetics world and human neuroscience research on fear and emotion and anxiety. For the past 2 decades, his lab has used molecular, cellular, and circuit analyses to study emotion and fear-linked disorders, often using mouse models. Dr. Ressler received his undergraduate degree at MIT and then did his MD and PhD at Harvard University. He was the first student of Nobel Prize winner, Dr. Linda Buck. We're working with her to help identify molecular organization of olfaction. He's a past president of the Society for Biological Psychiatry, amongst many other honors and distinctions, which I won't all read today. Before coming to McLean, he spent 18 years at Emory University in Atlanta, Georgia, where one of the fascinating things he did was found the Grady Trauma Project, which looks at intergenerational cycles of trauma exposure, PTSD, substance abuse, and depression in at-risk urban communities. And yeah, we're really, really excited to have you with us, Dr. Ressler. And thank you to everyone who registered and sent us questions and interest areas beforehand. I think Dr. Ressler is going to try to address some of them during the talk, but we also invite you at the end to turn on your videos, ask your questions. Definitely don't feel like if you wrote them in the registration form, you can ask them live. It would be wonderful to hear from you. And during the talk, also feel free to drop them in the chat box. So I guess
wonderful.
Get started.
Thanks so much, Parazad, for all that you do. The Harvard Brain Science Initiative is really just fantastic, and it's such a great opportunity for everybody in the community. And this learning series has been fantastic as well. Great to see a number of friends and colleagues in the audience, and look forward to meeting people I don't yet know. So when I first was talking with Parazad about this, I was thinking the time was much shorter, so I was going to give a very brief primer on fear. But then I realized we had close to enough time really for a pretty detailed talk as well as question and answer. So the way I'm going to break this down is an entry of sort of basic fear learning for anybody with some animal experience, showing how we do this in mouse models, and then sort of broaden that successfully to looking at some of what we know about sensory systems and fear and how that changes with fear. From some of the questions, there were fantastic questions in advance. Thanks, guys. And so I'll try to integrate a few things that I might not have talked about. Otherwise, I'll show a brief primer on some of our work on intergenerational transmission of fear and trauma and some of what we've learned about that. And then transition over to human disorders of fear, primarily focused on post-traumatic stress disorder, how we study that, some of what we know about the genetics, the neural circuitry, and then kind of end with some of the next step approaches, the translational understanding of neuroscience and fear, with one of the thoughts that this area may be one of the most tractable from a perspective of translational neuroscience and psychiatry. So with that, I will start, and please put your comments in the chat, and I'll try to stop where needed to clarify things, and then we should have plenty of time for discussion at the end. What we're particularly interested in is fear, and Parazad and I talked a little bit about at the beginning how I got into this. And I got into it because I was very interested in the neuroscience of psychiatric disorders. But as a basic trained neuroscientist and circuit neuroscientist, I really wanted to study something that I felt like was tractable within my lifetime. And I want to make the argument today that PTSD and human fear-related disorders are tractable for several reasons. Because one, we have a shared fear neural circuitry across species. Was talking with folks at the beginning about some of their work in fear processing in zebrafish even. Separately, particularly with post-traumatic stress, we know when it starts. Unlike almost any other disorder in psychiatry where we don't really know what the antecedent components are, by definition, PTSD starts at the time of the trauma, and understanding fear consolidation and fear processing is critical for understanding what happens in that disorder. And finally, it really relies upon the concepts of learning and memory, and independent of explicitly being interested in fear or trauma, using basic Pavlovian cue or context conditioning is used across cognition fields. And that then also allows us to take the over 30 years of great progress in understanding learning and memory and apply it to this specific set of circuits and questions and human disorders. And our goal is to have a molecular neurobiology of post-traumatic stress and fear-related disorders that spans understanding predictive biomarkers of intermediate phenotypes, brain phenotypes, genetic and epigenetic phenotypes that could lead to novel interventions based on the neurobiology. So very basically, an old picture from Scientific American from Joe Ledoux, one of the founders with Mike Davis and Mike Fanzelo and others, and really the modern neuroscience of fear and threat processing. I'm happy to talk about in the discussion the distinction between threat behaviors and fear behaviors. Briefly, Joe Ledoux would now say that fear is really a subjective emotion that humans express and feel, whereas anything behaviorally, we're looking at threat responses. And I'll try to use that terminology, but I'll probably mix them up a bit as well, whether I mean to or not. And the basic idea in this figure and some of his early work was that any given sensory experience can take sort of relatively rapid, he called it the low road, information from, say, the visual thalamus, from the first synapses from the eye, into the emotional, responsive, behavioral reflex regions like the amygdala that I'm going to talk a lot about. And versus the high road, where the same visual thalamic information goes to multiple layers of higher order visual cortex, higher order conscious processing, memory processing, till finally top-down regulation comes back to the amygdala. There's many hundreds of milliseconds between that very rapid sensory input into a behavioral reflex and that top-down regulatory component. And therein lies much of the problem, where we both have, from an evolutionary conserved perspective, the rapid fear threat reflex response, which keeps us alive from predators, but leads to dysregulation and disorders that I'll talk about. The main players on the human side, and really both across mammals, is again the amygdala as kind of the final common output, the medial prefrontal cortex, which has different components, but more the dorsal anterior cingulate and rostral anterior cingulate, we think is akin to the prelimbic region in mouse and seems to be associated with increased fear and threat responding, whereas the medial and subgenual cingulate that seems to be akin to the infralimbic in mouse seems to be involved in extinction and suppressing fear, whereas the hippocampus is involved in the contextualization, both contextual fear memories, the contextualization of any cue memories, but also learning safety, learning extinction is clearly hippocampal and contextual, and I'll talk more about that. So to put a little context in it all before I dive into some of the deep basic biology, is the concept of a panic attack for humans. And so anybody who's experienced panic attack or knows someone who has will hear something like, I felt dizzy, my legs gave out on me, I couldn't catch my breath, felt like someone was choking me, my heart was beating too fast, I thought I was dying. And what we now know about panic attacks is that they happen in a whole host of anxiety and depression-related disorders, and that many of the symptoms, from heart rate, GI distress, shortness of breath, chest discomfort, lightheadedness, choking, almost feel reflexive. And there's a long history of whether this is primarily brainstem mediated or amygdala mediated. But what we know is that while we might call them mostly associated with panic disorder, where they seem to come out of the blue, and which we now appreciate has more to do with generalization of threat cues and lack of cue specificity, they also occur in simple phobia. If you're afraid of spiders and if a spider lands on you, you have all of these symptoms. But we call it being scared or scared to death as opposed to a panic attack. With social phobia and PTSD, you see very specific panic attacks that are driven by the cue reminders of the speaking in public, of having a social interaction, of the trauma reminder, seeing a car if you're in a car accident. And that's starting to be well understood. What was exciting to me in my training, both as a neuroscientist in grad school and then as a psychiatrist, was appreciating from work like Mike Davis and Mike Fanzelo and Ledoux and others, that in parallel with the psychiatry understanding and representing the concept of a panic attack across anxiety and PTSD disorders, the neuroscience field had mapped out the amygdala mechanisms of fear responding, so that hardwired projections from the central media of the amygdala in mammals to brainstem regions, the hypothalamus, the dorsal vagus nerve, the parabrachial, as well as hypothalamus and the central gray, all are associated with many, if not all, of the hardwired, if you will, panic symptoms that we see in humans. So that essentially a reflex shown now with electrical activation in the central, or chemical activation, or now optogenetic activation in animals, as well as in surgery, for example, activation of the amygdala in human patients. People have shown that essentially you can think of this as a threat reflex response through amygdala activation, which then is quite exciting because that suggests more so than most any other area in psychiatric behaviors, we can really connect a specific set of behavioral responses to a specific set of symptoms and syndromes that we see in humans. And now we can start understanding what's upstream of that. So a lot of my talk in the field for the last 20 years is focused on how does this happen at the level of amygdala function, at conditioned stimulus and unconditioned stimulus connectivity, as well as other circuits, top-down regulators and part of the downstream regulators of this, and how does that go awry in disorders like post-traumatic stress disorder? So now to sort of back up to Fear 101, I guess, from basic Pavlovian conditioning. So again, Pavlov, we often think about Pavlovian work with some of the more well-publicized components of the dog with salivating to a bell. And of course, that's appetitive or positive learning, but equally well, and if not sometimes more quickly, animals can learn aversive learning. And particularly with fear conditioning, one can do it in one trial. And so from a learning and memory perspective, that's very exciting because single trial learning allows for very rapid time constraints on when the cue is presented and the unconditioned stimulus is presented, and thus to be able to examine the molecular components of memory consolidation. The basic idea is the conditioned stimulus is defined as a previously neutral cue. In animal studies, we often use a tone or a specific bite. This can be much more complex to a configural cue with many different components. It can be other sensory systems, air, etc. And in mice, again, we mostly use foot shock. In humans, we can use a brief hand shock. We can also use an air blast to the throat or even a loud noise as the unconditioned stimulus. The critical component is that generally the unconditioned stimulus co-terminates or ends at the same time of a conditioned stimulus, which is longer. And what we think is happening is that these neural circuits very rapidly are then combining these data so that in the future, the conditioned stimulus will be associated as a predictor of the co-termination of the unconditioned stimulus. That prediction then will now, after learning, such that the previously neutral tone or figure or experience is now associated with the hardwired, essentially switching the hardwired unconditioned stimulus responses. So the defensive behaviors, the autonomic arousals, hyperalgesia, reflex, and stress hormones, again, which all basically line up with these known hardwired downstream circuits of amygdala activation. The way we study this most commonly in mice and rats is fear-potentiated startle and freezing. Though increasingly, these are old slides I'm showing. Some of these are Mike's old slides from 30 years ago. But we're now also using things like deep lab cut, some of Bob Dada's approaches, and large, much more complex and powerful visual learning and machine learning tools to really get a better sense of the different kinds of behaviors. But I'm just going to mostly talk about these classic ones. So with classical conditioning, for example, in startle, and one thing that's interesting about startle too is it's one of the critical symptoms that we see in post-traumatic stress disorder, where people who previously had a normal startle response now have a hyper-arousal, hyper-alert startle response. The idea of a startle is that anybody who hears an unexpected noise like that, you might have a little bit of an eye blink, you might not. But if you were in a dark alley and you were in a position of hyperarousal and anxiety and heard that same unexpected startle, you would jump. And that startle response is mediated via the projections from the amygdala to the reticulus planus caudalis in the brainstem, mediating this muscular startle response. And the way we measure it in animals is essentially, in this case, the rat has been trained with the previously where the light was associated with the shock. Now you take the animal on another day, put them in a chamber, you play the startle sound in the dark, you play the startle sound in the light, and he startles more in the light. And you can measure that in a number of quantifiable ways. We usually use a piezoelectric component under their feet. You're measuring the vibration. And these are what the San Diego Instruments is one of the common people who make, groups who make the startle chambers. They usually with these, they have a little bit of room to move, but for the most part, they don't have a lot of room to move, and you're really getting a whole body startle response to the tone or the shock. Freezing, what we're doing is generally having a larger space. The idea of freezing teleologically is thought that when particularly a small mammal sees a prey, a bird, or another predator, that they want to be quiet. And so that this is essentially built in as this reflexive response. And what you'll see is they run to the corner and they're just completely still with almost no movement except for breathing. And the way this actually looks, so forgive the old video, I've got to get Emily in our lab to give me some better updates with some of our new deep lab cut stuff. But you can see these are animals that have been previously trained. The one on the bottom was not trained, the one on top was. And when the tone comes on the bottom, you see that, I'll start it again, you see that they both start exploring, but as soon as the tone comes on, the one on top runs and is basically frozen in the corner, and the other one continues to explore. And so with a number of video capture type tools, one can automatically look at freezing bins in 1 to 5 second intervals, usually over the course of a 20 or 30 second cue. And again, that's getting more sophisticated in how you can use these intermediate phenotypes. What this looks like at a data analytic level is with a paired, so where you do tone shock pairing, for example, you see much more fear potentiated startle and much more both compared at baseline, as well as when you're looking at percent increase in the paired animals compared to unpaired. And this is a common control for any kind of study that you're looking at with fear conditioning, because unpaired, you still get the shock and you still get the tone. So the animals experience all the same things, but they've not been presented together in associative learning path. And we know now through years of work that paired association leads to changes in the amygdala that are NMDA dependent, calcium dependent, BDNF dependent, and lead to long-term potentiation of synapses coming into the amygdala and cell activation within the amygdala. But that requires the association and not just the stress alone. In terms of how this looks over time, in this example, the mice were given 5 tone shock pairings, and we're just looking at the amount of freezing. Initially, there's very little freezing. As the animal in the paired condition continues to receive more and more shocks, they start to freeze more before each shock. So during the tones, you're essentially seeing this acquisition curve of the freezing over time. If you then look at a process we look at a lot with is extinction, and we think this is a model of exposure therapy, and is really a model of cognitive learning, hippocampal prefrontal top-down regulation of amygdala function. When after the animals have been trained, you're now testing them for the first time. They show a lot of freezing or fear potentiated startle. In this case, we're looking at fear potentiated startle. Those who previously had controls, they're not showing FPS. But over time, with repeated trials over and over again, that tone cue no longer predicts the fear, and the brain has now relearned that's not a danger cue, and we think that's mediated via prefrontal, hippocampal, and other circuits regulating the amygdala, and it's a plasticity activity dependent process as well. This just shows another way of looking at if the animals are exposed to the context but not the cue. During the extinction sessions, you'll show freezing initially that comes down, but importantly, now when you bring them back for what we call an extinction retention trial, another day or another week later, those animals that did not receive active extinction, if they're now re-shown the cue in the same context where they were trained, they have what we call renewal, high level of fear, whereas those that received extinction training show very low level of fear. They've maintained this extinction memory. Okay, so to shift gears a little bit, while we mostly focus on how this happens in the amygdala and the areas regulating the amygdala, what we've known for years is that when you do Pavlovian fear conditioning, you see changes in many other sensory areas as well. And this just shows, for example, a FOSS, an experiment we did early on in Mike Davis's lab with different immediate early genes in the aftermath of fear conditioning. And so this shows context alone animals, animals that received an unpaired tone and context. These were both at 30 minutes versus 30 minutes after paired tone shock pairing, 2 hours, 4 hours, and 8 hours later. You see this nice curve of a rapid window of immediate early genes, and other data suggests that you get a secondary wave as well several hours later. And this is, you see first a wave of immediate early genes, then you see a secondary wave of growth factors, and a second tertiary wave of structural change. But you see this not only in the amygdala, you see this in the auditory cortex, somewhat the auditory thalamus, somewhat the output regions. And so the evidence suggests that there's plasticity occurring in all of these regions. And this really fits on decades of work from other primary sensory biologists, Mike Merznick, John Cass, Mike Stryker, Weinberger, and others across somatosensory systems that suggest both developmentally, but also with salient learning, you can get structural change. Ori and Kiefer in my group a number of years ago showed that you can see structural change in the mouse auditory cortex that actually is detectable with structural MRI. And so this shows following multiple days of fear conditioning, he saw structural voxel-based morphometry increase gray matter density change in insula, the amygdala, the central amygdala, the medial amygdala, but also the auditory cortex. And what was nice about this mouse model is he could use a fluorescent reporter to actually count synapses. And he showed that this increased structural density that we see in some human studies of structural gray matter change with trauma over time is associated in this voxel-based morphometry model in the auditory cortex. So again, he's looking at gray matter density here. And what he sees is that gray matter density increase in postmortem analysis was associated with increased spine density with shoal analysis, suggesting that at least part of that gray matter density change, and we saw no change in number of neurons, number of nuclei, et cetera. So it suggested that at least part of that change is actually due to increased structural representation of the substrates of memory formation. Also in the auditory sensory cortex, Nina Banerjee in our group a few years ago was looking at structural representation of synapse function. And perineuronal nets is something that's been associated a lot with developmental change and with sort of closing the developmental plasticity periods. And yet Nina found that you actually, if you time it right, and look 4 hours versus 24 hours after fear conditioning. So these are adult C57 mice looking at home cage 4 hours and 24 hours later. These are all using stains looking at perineuronal nets. That she saw a significant increase dynamically in perineuronal nets in the auditory cortex at 4 hours, which had returned to normal by 24 hours. When she used various tools to block, like chondroitinase, to block or delete the perineuronal nets, she actually saw a blockade or a decrease in auditory fear conditioning. And the model that we've have yet to do further experiments on so far, but suggests that perhaps this transient increase in perineuronal nets surrounding inhibitory neurons in the auditory cortex actually leads to a transient disinhibition of these pyramidal neurons in the auditory cortex. And that then may support some of the longer term molecular and then structural changes leading to long-term plasticity. Some work areas that we've done a fair amount of work also in sensory systems with olfactory fear is the olfactory system. And what's exciting about this is we have different molecular tools or markers for each specific odor versus another odor. And what Seth Jones initially, and then Brian B.S. and Philomene Morrison in my group showed was that we could actually get a change in representation of the olfactory substrates in the nose and the olfactory bulb following olfactory fear conditioning to one specific odor versus another. So what we're seeing here, so again, if you're not familiar with the olfactory system, we know there are millions of neurons in the nose. They each project to the olfactory bulb, but based on one of approximately 1,000 different odor receptor genes they express, they aggregate and go to a very specific receptor-specific glomerulus. And this is LAC-Z that's expressed in specific neurons in the nose. You can see they're projecting to the olfactory bulb in an untrained animal or an animal trained with an odor that is not activating these. But if you train these animals to an odor that activates this specific receptor marked by blue here and follow those axons and wait a couple of weeks after multiple olfactory trainings, you see a significant increase in the number of neurons in the nose and the density of those axons into the glomerulus. And this just shows 6 weeks after training, seeing more neurons in the nose representing, in this case, M71, which recognizes acetophenone odor. We see that both in the neurons in the nose and in the bulb. And interestingly, if you then extinguish that odor over time, the percentage of neurons in the nose and the density of axons in the bulb returns to normal. So it looks like the whole sensory system can be dynamically regulated as a function of this salient learning process. Someone in the question and answer knew about some of our intergenerational work, so I thought I would just touch briefly on that as the next step. So again, we see in humans lots of evidence for intergenerational cycles of trauma, risk, and violence and behavior related to trauma and anxiety, both in psychiatry disorders as well as in social situations. And one example from some of our work over the years at Grady was if you take kids and look at their dark enhanced startle, so the startle response, but in a human's or their eye blink response, the kids' level of startle in the dark was highly predicted by the mother's level of abuse as a child. So very interesting observation. We see these kinds of observations a lot, that parental psychopathology at one level or another is high risk for kids having more risk. But how could that be? Well, it could be certainly via parenting and observation. It could be being in a shared risk environment, also be shared genetics. The question is there an epigenetic component as well? And Brian Diaz in our group started looking at this a number of years ago, and we were amazed by what we found. So this shows similar bulbs to what I showed you a minute ago. These are looking at the offspring of mice that they're C57s, but on a background in which we're seeing the specific odor receptor M71 that's sensitive to an odor called acetophenone. And if you remember this slide, so this is what it looks like if you take an adult and you train them to acetophenone versus don't train them to acetophenone. If you then look at the offspring of these males, fathers that had been trained to acetophenone, believe it or not, but the F1 offspring look like they have been trained to the odor. They have more neurons in the nose. They have more axons in the bulb specific to that odor and denser glomeruli. And this can be quantified in lots of different ways. They also show increased behavioral sensitivity to that odor. If you cross-foster so that it's not at birth, you're cross-fostering so that it can't be presumably behaviorally transmitted, you see the same increase over time. And if you go to the next generation, you still see this increase that's specific to one odor versus another odor based on which was trained. And this also shows the behavior in the F2 offspring. And fascinatingly, he was able to show, and I won't go into it, an epigenetic signature in the sperm that seemed to be associated with this. And then if we did full in vitro fertilization, so we trained the fathers, collected their sperm, sent it to the animal care facility where there was no acetophenone at all, so there's been no way it could be exposure related. They did IVF. Then we examined the offspring in a blinded way. Still, the IVF offspring had increased glomerular area and neurons in the nose related to that. So it suggests that that there is some level within certain types of sensitivity that can be epigenetically intergenerationally transmitted, though much work needs to be done. Notably, this main effect of intergenerational transmission of the olfactory bulb has been replicated to some extent by folks in Richard Axel's lab as well. So the interim summary so far is that classical conditioning procedures are commonly used to examine the behavioral and neurological controllers of fear. Like fear, inhibition of fear, which is called extinction, over time is learned. Compared to the production of fear, not much is known about, or not less is known about extinction. That's very critical for psychiatric disorders. Fear memory formation is and other sensory dependent emotional learning paradigms are associated with structural changes and increased sensory sensitivity, often in a sensory specific way. And at least some evidence is growing for intergenerational transmission of stress response, both in the cortisol HPA axis, as well as in at least some sensory areas like olfaction, leading to increased understanding of what may be happening, at least with some level types of intergenerational transmission. So I'm gonna keeping an eye on the chat. I don't think we have anything yet. So please, if you have if something doesn't make sense, jump in with the chat and I'll try to clarify, and then we should have plenty of time for discussion at the end. So I want to shift gears from this bit of a primer on how we measure fear, what are the main brain circuits in rodents, and what are some of the other ancillary regions in addition to the amygdala that we see, and particularly interest in potentially sensory sensitization that occurs over time with fear learning. Let's go now to humans and look at human fear-related disorders. So again, almost all psychiatric disorders, one can argue, have some interaction between genetic biology and the environment that one experiences. Post-traumatic stress disorder, we could argue, is one of the most quote-unquote clean gene by environment disorders, in that we know there's genetic underpinnings. It turns out the genetic heritability with large-scale SNP models is about 30 to 40%, which is not that far off of where we are with depression and bipolar, with schizophrenia and autism being the most heritable, more like 60 or 70%, and mood and affective disorders and PTSD being more like 30 to 40%. But that said, even things like schizophrenia and autism have some level of environmental exposure. How much of that's behavioral, how much of that's toxin, or many other things. But PTSD, because we understand almost by definition what the environmental component is, allows us to understand this potentially in a more powerful way. Again, what is it? What do I mean by fear disorders? Well, more clearly within the broad nosology of anxiety and stress disorders, post-traumatic stress, phobias, specific phobia, panic disorder are all things that are very clearly associated with fear responding. Though one could argue that most anxiety and OCD have components of this too, and they certainly have shared neural circuitry. In terms of how does this overlap with memory? Well, again, we can think of the memory systems being broad, from sensory memories to working memories to emotional and contextual memories. And all of these interact together to regulate a number of components. And while I'm talking today about the role of amygdala in fear, amygdala has an equally important role in appetitive behavior, in learning, in sexual behavior, in stress responding, and even in attentional responding. So I'm just telling you a tiny slice of the story. PTSD, for those who aren't as familiar, again, is clinically important. Up to 5 to 10% of the population can be affected. Highly traumatized populations like veterans and highly traumatized civilians, it can be as high as 30%. It's a significant cause of suicidality and disability and comorbidity with both depression, other disorders, and substance abuse. What it requires having been exposed to death, threatened death, or actual serious injury, in sexual violence, and it usually involves significant intense fear at the time of the trauma. And the clusters of symptoms are intrusion, so re-experiencing through nightmares and flashbacks these memories, avoidance of things that remind, avoidance of those cues that were traumatized. Cognitive and mood symptoms is a new addition from DSM-4 to 5, but it really contextualizes the idea of decreased cognitive function. And what we think is that when the emotional hyperarousal systems are hyperactivated, many of the more top-down regulatory executive functioning areas do not function as well. We'll talk more about that if you'd like. And then arousal symptoms, of course, one of the classic components of PTSD is increased arousal, decreased sleep, hypervigilance, increased startle, increased stress responsiveness. How do we treat it in one slide? So from a medication perspective, there are actually no specific medications for PTSD to this day. There's only 2 FDA-approved drugs, and that's sertraline and paroxetine, Paxil and Zoloft. So again, those are our standard antidepressants. It turns out that almost any antidepressant also works at some level for PTSD. So it doesn't get specifically at the memories, it just dampens the overall symptom response. One of the most interesting drugs in PTSD is prazosin, which is a specific alpha antagonist. So that's the closest we have to probably a somewhat commonly used specific drug for PTSD. And for a subset of people, though we still don't quite know who that subset is, it seems to work very well to decrease nightmares and decrease nighttime hyperarousal through decreasing adrenergic activity. And where the field kind of is right now is trying to identify what are better biomarkers that predict who's going to respond to that. And what's fascinating is we have such a good neuroscience of the area yet, and yet there's still no treatments based on that. And certainly that's something we hope that in the next 5 to 10 years will change. Our best treatments really are still the targeted psychotherapies. We're essentially retraining the brain, and the types of therapies are prolonged exposure, cognitive processing therapy, cognitive behavior therapy, but they all rely in many ways on 2 basic foundations. One is sort of skills training for emotion regulation. 2 is really psychoeducation about understanding the biology of PTSD and what's happening and that you're not going crazy. And giving tools for top-down emotion regulation, the cognitive part. Then the final part that's really seems to be therapeutically critical is exposure. And what happens with exposure therapy, if you look on this graph, for example, is this is your level of subjective anxiety. And the first time you tell the therapist your story of your trauma, your anxiety goes through the roof. But if you still keep staying there and don't dissociate, talk about that, and don't run away, don't avoid, over time in that session, that memory will become more tolerable and you'll be less anxious. The next time you do it, it will be a little bit less, the next time a little bit less. Essentially, you're making that overwhelming emotional black hole of trauma memory more just like a boring bad memory. And this is effectively the exact same thing that we see with extinction of fear in model systems or in human preclinical studies. And so understanding extinction of fear is critical for our understanding of our basic treatments for post-traumatic stress. I've talked about the Ledoux model. Again, the amygdala is conserved across mammals and even to some extent, at least in a vestigial form, across all vertebrates. Let's see, chat. Important, don't have time to talk about all the components, but there's definitely, TMS, we're having increased evidence for PTSD, whether that's really targeting the PTSD memories or more related to decreasing general negative symptoms. Oxytocin is an interesting, which in the discussion can talk more about. It may be helpful, though there's some evidence that, depending on someone's background and their trauma type, et cetera, that it increases general sensitivity to social interactions, but for someone who's been socially traumatized, sexually traumatized, it can actually make some of that sensitivity worse. So I think that remains somewhat complex. And I'm going to talk a lot about some of the other ideas about types of treatments as well. But again, the idea that fear, while it evolved to protect us in modern day humans, leads to increases in many of these disorders and symptoms in a way that is probably much more harmful than helpful. Diving a little bit more into what the circuitry. So I talked about the hardwired amygdala output before. Again, we think this is modulated by hippocampus, prefrontal cortical, and other areas, as well as sensory sensitized inputs coming in. What we think happens is during the actual trauma formation, the conditioned stimulus, previously neutral, is paired with the unconditioned stimulus, the trauma experience in the lab. That's the shock. In real life, it's the car accident, it's the assault, it's the pain. And that via these structural changes that occur in both glutamatergic, calcium-dependent, LTP-like changes, in the future, the conditioned stimulus alone is able to activate the hardwired fear circuit we talked about. So from a overall learning and memory process, we think about PTSD in a broad way. The big question is to, and I think one of the more interesting ways to look at it is to flip it on its head. Not why do we develop PTSD, but why, given a significant trauma, to only a subset of people develop PTSD. So given any given trauma, we only expect 5 to 10% of people to develop PTSD, and the vast majority over time will recover. What's the difference between those two? Well, one is the genetic sensitivity that I'll get to a little bit, that we're just starting to understand more about from a large-scale genetics perspective. The other, of course, is environment in many different ways. And we know childhood trauma exposure is one of the biggest risk factors for all aspects of psychiatric disorders, but certainly within PTSD, it looks like early developmental trauma sensitizes this amygdala fear system so that later adult trauma then responds in a more significant way. What happened in the immediate aftermath in the days and weeks, that learning of consolidation of fear, I'll talk quite a bit about, but we think that's a critical differentiator. And then with chronic trauma symptoms, PTSD symptoms, one has ongoing intrusive memories, nightmares, flashbacks, and behavioral and cognitive responses. Those who recover tend to discriminate. So may say, I was attacked down this dark alley, but that was that place and that time, and I'm okay now. Whereas the person who has PTSD may say, they generalize these fears, these contexts and cues. And they say, I was attacked down this dark alley, but now I'm kind of afraid of that general area. I was attacked by a man, but now I'm kind of afraid. I don't really want to be around men in general. In fact, I don't really want to be in that part of town. Now I kind of don't really want to leave my house after dark. Now I kind of don't want to leave my house or my bedroom. And so the world becomes more and more dangerous and less safe as threat and fear memories are generalized. And we think that's very highly hippocampal dependent, that generalization versus discrimination of cues and contexts. Similarly, those who have PTSD will say, Doc, every time I think about it, every time I have a nightmare, it seems to get worse and worse and worse. And even when I talk about it, I just don't want to talk about it anymore because it gets worse. And that's this idea of sensitization clinically. That's in contrast to the concept of extinction. And extinction, again, as we talked about therapeutically, is when you talk about it in a therapeutic environment, but we're actually extinguishing all the time. Anytime you're talking to your friends about what happened, or you're kind of just thinking about it and you're not escaping from it, you're just trying to come to terms with it and you're thinking about it again and again, it slowly extinguishes. You're relearning that I can now have this memory, and the memory does not have to be dangerous. Whereas with PTSD, it seems that the memory and the present get conflated and it always feels dangerous. With sensitization, we're also thinking about this relatively new concept in memory with reconsolidation. So anytime a memory is reactivated, does it have the possibility of being sensitized and strengthened or reconsolidated, versus is it diminished in its representation, either through things like LTD at the same local synapses or through different circuits related to safety learning, is it extinguished? So that's a constant give and take in the memory field, and what may be happening in real time anytime a memory is activated. One person asked, can you describe associative learning via trauma and extinction for things like OCD? And we don't know as much how trauma directly relates to OCD. We certainly see it. One way I think about it is that OCD is very much, and I would think I'm not going to show a slide on this today, but one of the processes we see across disorders is this repetitive habitual behaviors. And Addiction is the place where we best understand addictive behaviors, and that's what we think is lack of top-down regulation of have this cue, you have this stress, you go towards this previously appetitive behavior, but now it's in a habitual and not a goal-directed way. OCD, again, is habitual behavior. Anytime there's the obsessive thought, there's the compulsive behavior to do this thing to decrease the stress of the compulsive thought. With fear disorders, I think you can almost think of it as you're in the habitual responding of avoidance, of a threat response, of an intrusive memory. You're not able to break out of that habitual aversive response into a different kind of non-aversive response. So all of these seem to be related to top-down, be they prefrontal or orbitofrontal, top-down regulation of amygdala and striatal habitual responding. And it's likely partly genetics, given early trauma, why one is more likely to go down one path or another that we don't understand so well. So I'm going to talk more for most of the next part about trauma consolidation and how we understand that broadly in the early aftermath. And I think that's particularly exciting because in more so than almost any place in psychiatry, we have the ability in PTSD potentially not just to understand it, but actually to prevent it. Because if we can understand what happens in those early hours and days in the aftermath of trauma and do something about it so that the long-term trauma memory was not overwhelming, so to help promote a normal recovery process, we can actually prevent PTSD. And we envision things like in the same way you go to the emergency department with chest pain. If you get a TPA or intervention early enough, you can prevent the long-term damage of a heart attack. Could we do the same in a cognitive learning and memory way to prevent the long-term damage of emotional trauma from developing in PTSD in the emergency department or on the battlefield? I'll briefly touch on some of the new large-scale GWAS and genomic stories related to PTSD, and then talk a little bit more about how we can use mouse genomic tools to better understand next generation approaches. And finally, by understanding extinction, how we might also use neuroplasticity related to tools to enhance extinction and therapeutic extinction in recovery. So again, consolidation, the idea has been around for a long time. Jim Magad Irvine's one of the earliest thinkers in this area about how memories go from short to long term, with the idea that there were likely molecular substrates mediating short to long lasting. And I saw Mike Greenberg maybe on earlier, and obviously Mike has been an international leader for decades in this area at a molecular level as well. Mike Davis, who is my mentor, really worked out a lot of the circuitry and behavioral component of amygdala-dependent fear. And in his lab, I did some of the earlier molecular analyses of amygdala activation in the aftermath of trauma. And where this leads to is a modern model in which the traumatic event leads to a short-term memory, but then is processed through over consolidation to long-term memories. And that this concept of an engram, I think Sheena Jocelyn and Susuma Tonegawa and others, that engram is really part of this whole network: the early sensory, the late sensory, the thalamic, the amygdala, the hippocampus, and the output pathways. And all of that is now sensitized in this way, if you will. And that when that memory is reactivated in a stressful way, it may be strengthened and reconsolidated. When it's reactivated in working memory, however, and held onto and not responded to in a as much of a stressful way, when other tools are brought in to help calm one down, and most importantly, the unconditioned stimulus, the predicted negative outcome does not happen. So there's this negative memory signal, extinction then occurs, and that can then lead to inhibition. But again, if we can understand this early consolidation phase, we may be able to have early preventions. So we and others initially at Emory in Miami with Charlie Nemiroff, and then now through a much larger study called the Aurora study with Sam McLean, North Carolina, Kirsten Conan, and Ron Kessler at Harvard, have been studying now many thousands of people in the early aftermath of trauma in emergency departments and collecting blood-based biomarker data, physiological data, and neuroimaging data in the hours to days to week after human trauma to start to understand what are the predictors of PTSD and where might there be targets that we could intervene. And from a neuroimaging perspective, again, just chronic PTSD, it's just one of many data sets showing that if someone with PTSD is passively viewing fearful faces, they see they have a more amygdala activation response to fearful cues versus neutral cues compared to trauma controls. And in parallel with that, we see less top-down regulation. So if we look at amygdala functional connectivity, you see less medial prefrontal functional top-down regulation of the amygdala. That's part of sort of the canonical fear circuit. Jenny Stevens in our group showed that this early amygdala activation in the first weeks after trauma can be a predictor of later PTSD. So we looked at people that we followed for up to a year, and between 2 and 4 weeks after the trauma exposure in the emergency department, we brought them back and looked at amygdala and anterior cingulate cortex activation to fearful faces. And the take-home message is that this activation signal, again, a few weeks out before we can even diagnose PTSD, which you need to have 1 month for PTSD or 3 months for chronic PTSD, this few week out acute stress amygdala activation was predictive of PTSD symptoms at 3 months later and 12 months later with chronic PTSD, suggesting that these early sensitization processes are already there. Now, whether they're pre-existing the trauma or whether this is the early process of consolidation remains to be seen, but some of the data suggests that at least in a subset of these people that didn't have high symptoms before this trauma, and that this is an early sensitization process that we're seeing. In contrast, remember the hippocampus and the medial prefrontal cortex are more serving as top-down regulators, and generally we see greater hippocampal functioning, greater hippocampal volume associated with more recovery, more extinction. And Sana Van Roy showed, again, in this early aftermath, so 2 weeks after, 2 to 4 weeks after the trauma, hippocampal activation to a stop signal task was inversely associated with later PTSD. So higher hippocampal activation on the x-axis was associated with much less PTSD 3 months and 6 months later. And one of the questions, an example, was about biotyping. And so we're seeing some of this in schizophrenia and depression. This is some of our early work and some of the first PTSD work on this, where we've taken several hundred people in the 2-week period after trauma and said, can we let the brain signals help us to identify subclusters of different types of biological PTSD, even if their symptoms all look the same? And Jenny found 3 broad clusters, and we think as the sample sizes increase, we'll see more clusters and have more fine granularity. But the first one she labeled reactive disinhibited, and that seems to have a lot of anterior cingulate activation, but also a lot of brainstem activation. So very, very reactive, very startle reactive. The next group, she said, had low reward, but very high threat responsiveness. And as we might predict, that was very driven by amygdala activation and insula activation. And then the high reward group, and again, this is all using the input data was functional tasks with resting state fearful faces, resting state reward behavior, and stop signal inhibition. But the high reward was more amygdala, ventral striatum, and hippocampus. So that looked like this broad group of people who develop PTSD symptoms could be broken down into biological subtypes. And interestingly, using our current symptom measures like the PCL5, we don't see any difference in their overall symptoms, and yet you're seeing these biological subtypes. So this is early data, but it seems to be tracking into some of what's seen with other kinds of behavior. Laura Payne just asked about sex differences in estrogen and progesterone, and there's definitely we see, I don't know if I mentioned this, about 2 to 1 female to male ratio of PTSD to males. While we see more sexual violence in females and more PTSD as a trauma type, even when you control for sex differences in trauma exposure, you still see an increased effect in females. Generally, it looks like higher estrogen and progesterone are more protective. And so that both with extinction, higher estrogen and progesterone are associated with greater extinction and less fear, and lower estrogen and progesterone are associated with more fear and less extinction. But a lot of that still needs to be done. And Mo Mabad, who's really a leader in many of these areas, has also done a lot in sex differences. Thanks, Laura. In terms of potential rapid physiological predictors, we can also see that adrenergic system responding. So basically galvanic skin response, a classic psychological tool where you're essentially just looking at sweat levels in the fingers. And we think this is one of the main output measures of the sympathetic adrenal response. And we're asking people, we're looking at people's galvanic skin response in the emergency department in the first hours after trauma, often while their stills taped down to a board waiting for, say, their neck spine, C-spine to be cleared or something. And we simply ask them, after we've, of course, consented them, what brought you into the emergency room? And their galvanic skin response to this open-ended question about what brought you into the emergency room very robustly predicts their PTSD symptoms 6 months later, whether we look at it categorically or continuously. This now has to be replicated in other ways, and we're starting to replicate it in larger components. But it's very intriguing that early sympathetic and HPA axis and blood-based markers, and we're just starting with large-scale RNA, that we're seeing some non-coding RNA effects as predictors, and some broader biomarker effects and inflammatory effects. They're not quite robust yet, but happy to talk about that in the discussion. So moving from consolidation to just so what's happening immediately after? I'm going to move a little bit to, well, who is differentially at risk and what do we know about the genomics? See, one other question by Nancy in the chat was that fear and safety signals are associated with changes in brain rhythms. Certainly, we're doing a lot in the areas of EEG and starting to understand more about EEG biomarker predictors, as well as rhythms from like a circadian perspective. But I don't have any good answers for you yet in terms of why that may differentiate resilience versus not. But I think it's a great question. In terms of the genomics of PTSD, there's been a lot in this sort of pre-large scale genetics literature on certain pathways, particularly focused on the hypothalamic pituitary adrenal system. FKBP5 is a pathway that we know regulates glucocorticoid response and has been quite associated with gene by environment differential sensitivity to trauma leading to PTSD. Also associated with increased amygdala activation, decreased hippocampal functioning, et cetera. And PACAP is another pathway that's upstream of the corticotropin-releasing hormone system and is associated with trauma and PTSD and seems to have a sex difference as well. Larger scale genetics, initially with, so I'm one of the co-directors of the Psychiatric Genomics Consortium, which is also co-led by Kirsten Conan at Harvard and Caroline Nevergelt and Murray Steen at UCSD. This now has over 200 collaborators across the world. And the first find, and it's rapidly grown. So PTSD was one of the areas that had the least genetic data 5 or 8 years ago compared to, say, depression and schizophrenia. Now it's rapidly catching up, and we really are very close to having a robust genetic architecture of PTSD. The first paper 2 years ago showed that we had robust genetic heritability that was on the same order of depression and schizophrenia. The most recent paper from Millions Veteran Programs and the InPress paper from the Psychiatric Genomics Consortium 2.5 are showing somewhere between 10 and 15 GWAS hits. And Freeze 3.0, which is due to be finalized this summer, will have on the order of 60 to 80,000 patients with PTSD and about 300,000 total. And that looks like it's going to have on the order of 30 to 50 genome-wide significant hits. So really starting to have some robust genetic architecture, which is very exciting. One of the most exciting early findings, and this comes from Murray Steen and Joel Galertner's work in the Million Veterans Program, is that one of these strong hits is in a chromosome 17 locus. And it's a region that has an inversion and has a lot of interesting genes, some of which also are associated with Alzheimer's disease. But importantly, many of these top polymorphisms are associated with corticotropin releasing hormone receptor 1. So again, CRH-R1 is probably one of the best understood genes in the corticotropin releasing, the hypothalamic pituitary adrenal stress axis regulating cortisol response. So while in no way is this the only story, it's very exciting that early on in the genetic architecture of PTSD, we're starting to see signals that map onto our understanding of the stress response. It's kind of like when schizophrenia, around number 50 or 100 in their GWAS found the dopamine receptors, we're validating some of the longstanding work in the biology of the field. Let's see what the chat. Does higher level of progesterone during pregnancy aid in PTSD treatment in terms of PTSD inhibition? It's a great question. There's some ongoing pregnancy and PTSD studies. I don't think there's anything definitive to say yet, but it's a good hypothesis. Okay, so I want to shift gears now into how do we go from human genetics, and I can talk later more about what's understood more about some of these gene signals already, but to using modern day genomic tools to understand more about the molecular biology of development of PTSD and risk. And so one standard approach, again, as we use in a lot of different studies, to do now genome sequencing or RNA sequencing. In this case, we're doing RNA sequencing to identify novel pathways associated with fear conditioning at 30 minutes or 2 hours after training compared to home cage controls. And one of the top genes that we found was called tachykinin 2 or neurokinin B. What was nice about this is as we see with many other plasticity genes in the aftermath of trauma, we see this dynamic increase that then returns to normal within a few hours after fear conditioning. And we see it only with associative learning compared to non-associative or non-paired learning. What was particularly exciting about this particular gene was its location. I talked about in terms of the central amygdala output as kind of the final common pathway of hardwired projections down to the brainstem, but not a whole lot was known about markers of the central media. PKC delta is a gene for which we know a fair amount about, and that's expressed in the central also been associated with regulating fear responding, as well as enkephalin, CRH, and others. The centromedial, we had more of a dearth of markers, and TAC2 was expressed specifically in the centromedial, and as shown here, well, that's the PKC deltas, TAC2 is in the centromedial, and David Anderson's group also showed it in the medial amygdala. What was nice is so it's got a good location, it's dynamically expressed in the aftermath of fear conditioning. Is it related to fear consolidation? It turns out it is. The third thing about it that's nice that it already has a well-established drug that's actually safe in humans called a sanitant. And if you give this drug, a sanitant, 30 minutes before fear conditioning, 10 minutes after, or 1 hour after, but not 24 hours after or 4 hours after, you see a significant decrease in fear consolidation in the aftermath of trauma. Again, coming back to how can we mediate trauma consolidation. If we give the same drug before fear conditioning and look at other molecular markers, such as the PACAP receptor, which is dynamically increased with fear conditioning, we block that, so it seems to be upstream of other markers of fear consolidation. If we give it specifically within the amygdala with cannula in the aftermath, so you do Pavlovian fear conditioning, give the drug, test the animals later, you see significantly less fear in those that had the TAC2 blocker compared to those that did not. And if you genetically, instead of blocking it, as we did with the antagonist, you genetically overexpress tachykinin 2 in the central amygdala, you get an increased fear consolidation, increased fear expression days and weeks later. And finally, you can combine these experiments and do an experiment in which we have a 2x2 design. We either genetically overexpress it with lentivirus or versus GFP, or we give the antagonist versus vehicle. When we give the antagonist with the control virus, you see a decrease in fear consolidation. When you give the virus alone, you see the aforementioned increase in fear consolidation. When you give the antagonist plus overexpression, you normalize the fear response, suggesting we can push and pull it in both directions. So finally, how can we take our knowledge of plasticity to enhance fear extinction? And again, I mentioned that our clinical exposure therapies are basically reactivating the memory over and over again until it has less and less and less salience or strength. And this was defined as long ago as Pavlov. We know it's amygdala dependent. We know that it's from Mike Davis's work years ago that it's NMDA dependent. Particularly Bill Falls, who now is at Vermont, showed that learning to inhibit fear extinguish fear is specific glutamatergic and NMDA dependent and activity dependent. What they showed here in rats is if you train rats to be afraid of lights and then test their fear. So in this case, what they've done is train the animals to be afraid. And then they did extinction of these lights. So they gave them 60 lights in the absence of any shocks, and you get this normal extinction curve. So now you show very little fear or fear potentiated startle. You retest them. If, however, you do the training, the 60 lights in the absence of shock, in the presence of a classic NMDA antagonist, AP5, and then you test them again without any drug on board, you still see as much fear as ever before. So you don't learn to extinguish fear if you're blocking NMDA, and it's been shown in a number of different ways. A couple, I guess, close to 20 years ago now, we showed that if you do NMDA activation with a partial agonist, D-cycloserine, you can actually increase fear extinction rate. So in this case, we did a partial extinction with 30 lights instead of 60, and we get a partial decrease in fear. But if you do that partial extinction now with an agonist of NMDA receptors, you get a significant increase in extinction of fear. And we then showed in humans that you could do this and enhance exposure therapy in a virtual reality exposure therapy situation for fear of heights. And that was replicated in a number of different exposure therapy paradigms for social anxiety, for OCD, for PTSD, and for panic. And initially the data looked very good, very exciting. Okay, we can use cognitive enhancers specifically to enhance exposure therapy. And however, then some of these data were not replicated. And then in large scale studies, it looks quite complex that there's a subset of people who respond and do better, but then there's others who don't. And what we think is happening, and one of the groups, Adam Gostella, showed that only those who showed good with in-session extinction and had D-cycloserine on board showed good with between-session extinction, suggesting from other studies that Richard Thompson and others showed that if you gave D-cycloserine, you could actually both enhance reconsolidation and enhance extinction, depending on what was the prevailing memory at the time of trauma. And so it suggests that simply giving plasticity is not enough. And this, we can talk about ketamine as another plasticity enhancer that people are using in different ways. But you actually have to have behavioral tools or know the right people where you can drive the proper learning that you want to drive, so that you're not just enhancing don't have the risk of enhancing the fear, but you're specifically enhancing the extinction. And part of the new insight into this is the fact that there are actually different types of cells within the amygdala that seem to separately encode the fear response versus the extinction response. So this work from Cyril Harrian and Andreas Luthi showed, when they did recordings of amygdala, that some cells responded to tone, did not respond to tone at all. Other cells did respond to tone, but only responded to tone after fear conditioning. So when the tone represented the fear, the cells responded strongly. But when the tone was then extinguished, cells didn't respond. There was a separate set of cells that didn't respond to the tone alone. They didn't respond after tone fear conditioning. But intriguingly, after extinction, when the animal behaviorally no longer showed a fear behavior, those cells now were activated. A lot of further data suggested that there are fear on versus fear off, or threat on versus threat off, or repetitive behaviors that are differentially modulated at microcircuits in the amygdala. In one example, Kerry J and Susumu Tanagawa in different kinds of studies have suggested that the cells that project from BLA to central amygdala may be the fear-on cells, whereas cells that project from BLA to accumbens may be the appetitive on or extinction on cells. And some of our take-home messages, I've talked about it, the amygdala is a broad thing, and then multiple subregions of the amygdala, and now we know there's hundreds of subtypes. And how can we use modern tools to get at the subtypes within the amygdala? I'm going to go ahead and finish in the next 5 minutes, then we'll start questions and discussion. So one way, area that we focused on is a subtype called the Thy1 cells. And Thy1 is a developmental marker, but it turns out it's expressed very specifically in adults only in a subregion of the whole temporal lobe, and specifically within a subset of pyramidal neurons in the basolateral amygdala. And if you look at CAM kinase as a pyramidal neuron marker, the Thy1 cells are only expressed in about a third to a half of those. It turns out if you optogenetically stimulate the Thy1 cells, you impair the traditional lateral amygdala through BLA through central amygdala feed-forward system. And when you impair that system and you combine activation of the Thy1, what we are now calling fear-off cells, with the classic fear conditioning model, you actually prevent fear consolidation. So if I train the animals to be afraid, then you either can show you different studies, but you can either prevent fear consolidation or you enhance extinction. In this case, we enhance extinction exposure. If you do that chemogenetically, so we train animals to be afraid, then we chemogenetically activate them during fear expression or extinction, you see much less fear. We can do all this in the opposite. We can do halorhodopsin or a GI dread, and we get increased fear. So in both directions, it's like you can push and pull fear consolidation or extinction consolidation by manipulating these neurons. Kenneth went on to do single cell sequencing of these specific neurons and identified a whole host of different gene markers of these specific cells. And just as proof of concept, we asked, could we take some of these markers that have known targets, known druggable targets, and hit them? And one of those markers is the neurotensin receptor 2 in TRSS2, and Kerry J has done some work on neurotensin as well in this area with consistent results. And what we found is the neurotensin receptor in the amygdala overlapped by one receptor. And there was a number of drugs, in this case, the agonist, beta-lactotensin, that we've never thought about before. We hadn't thought about neurotensin 2 receptor 4. But neurotensin 2 is a GS-coupled receptor. And our argument was if we gave an agonist for the neurotensin 2 receptor in a cell population that we predict is a fear-off cell population, we should decrease fear and enhance extinction. And we got one of the most robust effects I've ever seen. This just shows the data of decreased fear expression and enhanced extinction in animals given beta-lactotensin. It was purely predicted by knowing the molecular architecture of the amygdala cells. So what that suggests then is that as a field, we're starting to get closer to both understand the consolidation process, understanding microcircuit consolidation and the molecules that are involved. And through that, we might be able to start having interventions that are targeting molecular consolidation, that are targeting cell and circuit processing, that are targeting extinction, so that over time, we might be able to have neurobiologically-driven approach to either blocking consolidation in the first place, knowing those who are at most risk, having intermediate phenotypes to see who's more likely to develop PTSD or not, and for those who have chronic PTSD, use neurobiologically-driven ways to inhibit fear and enhance extinction of fear. So I'm obviously just, so this just sort of further summarizes that, such that we hope that that there can be a really tractable set of disorders based on fear, and that precision medicine could be used for new interventions and preventions, as well as providing much greater understanding of the neurobiology of specific behavior. So I've tried to thank people and call out the various postdocs and students along the way and collaborators. It's very much been a team effort, and I'm really just the cheerleader of a great team. Thanks so much for your attention, and I really look forward to discussion and question and answer.
Thank you so much for that fabulous talk. So please, people, feel free to raise your hands, turn on your video, or type questions in the chat box.
So yeah, so it looks like one from Joy Otten. Thanks, Joy. The question is, do we know what cell types respond to fear conditioning and which ones do not? And the answer is sort of. There's a few examples where we have quite a bit of data. The early Luthi paper that I showed was pure blind physiology. Now with single cell sequencing and things like cell type specific GCamp neuroimaging, we're starting to get a much better idea of the cell types. And so we're starting as a field to have, being able to put together, okay, these are the markers that seem to robustly across labs predict fear on or fear conditioning. These are the markers that robustly predict inhibition. Those are still relatively few. We have a lot of markers in cells that we still don't quite know the answer to, or they might be active in one paradigm and not another, and we're not sure. But more to follow. I think Kara Weissman had her hand up next.
Yeah, hi, thanks. I'm the one who asked about the intergenerational work in the forum, so thank you so much for addressing that. I mean, I'm sure everyone's wondering, like, I found the paper super convincing and it's an amazing result. Do you have any follow-up work on what the mechanisms are? You must get this all the time.
Well, the most is for terms of the mechanisms. Brian went on to UCI. I moved to McLean, and we haven't quite done as much as we'd like to yet. It's a bit behind. Our favorite hypothesis is two follow-up papers. One, a paper from Brian where they fully replicated the effect, and we also fully replicated it at McLean. So it's been done in a couple different sites, and then I'm blanking on her name. But a fantastic junior faculty member in Richard Axel's lab has also replicated the main effect, though it's not published yet. But Brian's data suggested that if you fear, I'm sorry?
I think it's Bianca Jones-Marlin.
Bianca, yes, Bianca Jones. Thank you so much. Apologies, yeah. Bianca. Yeah. And Brian had showed that if you do fear conditioning in the adults and then extinguish the adults before mating, you don't get the intergenerational effect. So to me, that's the most exciting take home, is that while this may be sort of faded, if you will, we can treat it. And if you treat it, you may block the intergenerational cycles of risk. So that's one hope. As far as mechanism, my favorite hypothesis, for which I have very little data, is that piRNAs can be broken down from mRNAs, and each odorant receptor is chock full of each olfactory sensory neuron is chock full of specific mRNAs for that receptor, and likely has piRNA for that expressor. And then piRNAs can be put into exosomes, and there's a whole literature in C. elegans and Drosophila that you can have cell type, you can transmit epigenetic effects via piRNAs, via exosomes. So I think all the parts are there. We just haven't explicitly showed it. Tracy Bale's work has probably shown some of the most in intergenerational transmission in ovaries, but happy to follow up with you offline. There's a lot of other data that I didn't talk about, but we don't know the specific mechanisms with this yet.
Cool, thank you so much.
Farzad, I'll let you help me call on people because I haven't been paying attention to who's next.
So let's kind of alternate. So I will read out a question from the chat and then call on people with their raised hands. So Yael has a question. What are your thoughts on complex PTSD? Does work in the field suggest that there are unique molecular or cellular underpinnings of this versus more standard PTSD? Do you know whether the effectiveness of various types of treatments is similar between PTSD and complex PTSD?
Great questions. I could talk for several hours about it, so I'll try to do a 1-minute answer. And so complex PTSD, for those not in the field, seems to be somewhat of a mix between post-traumatic stress disorder and emotion dysregulation disorders like borderline personality disorder. It seems to be most common in people with developmental trauma, childhood trauma, as well as ongoing adult trauma, so multiple traumatized over time. And it seems to be a real mix of emotion dysregulation as well as interpersonal difficulties and rejection sensitivity with more classic PTSD symptoms. It seems to respond best to, as you might imagine, approaches that address both, a combination of things like DBT, dialectical behavior therapy, focused on skills training for emotion regulation, combined with, over time, more traditional trauma-focused PTSD therapies. I'll maybe pause there for the treatment. Molecularly, we have no idea yet similarities or differences. Simplistically, as a neuroscientist, the way I think of it is the more trauma one has, particularly during development, the more healthy development is disrupted and the more systems that go wrong. And so if you have trauma that's partly related to child maltreatment and interpersonal development and proper emotional development, one is more likely to have disruption of those systems in parallel with later trauma that associates more of the hyperarousal disruptions. Happy to follow up offline, but there's a lot much more we don't know than know at this point.
So I think Iris has her hand up.
Yeah, hi. So first of all, thank you for this really great talk. My question is also about this transgenerational work. Did you try or do you get fear generalization or extinction deficits in the offspring at weak protocols that you would not usually expect such phenotypes?
So it's a great question. I mean, if the hypothesis is that our favorite hypothesis is that it's not truly fear you're inheriting, but it's the sensitization of a specific sensory system. If that were the case, we should be able to show that in other ways that haven't been done yet. Isabel Mansui in Switzerland with Tracy Bale have done more showing just sort of stress interaction and changes in stress responsivity over time. It seems to be very much HPA regulation. And so it may be that you inherit multiple things kind of in parallel. Maybe you inherit certain sensory sensitivities and you inherit, based on your trauma experience, some level of increased stress preparedness, and that maybe those two together look more like a specific fear conditioning. But again, there's a lot more we don't know than know. And I don't think we've done explicit experiments about generalization and sensitization that you might suggest. So great ideas, haven't done it yet.
Thank you.
Mm-hmm.
So another question from the chat from Nancy. Can you tell which interneurons are involved?
Again, there's so much literature now on this, it kind of depends on which question.
I won't ask you now. I'll try to talk to you at some other point.
Yeah, I would love to follow up with you. There is a pretty good literature on transient decreased GABA activation in the amygdala during consolidation, and that's consistent with the hyperexcitable LTP mediated process in the amygdala.
There's reason to believe that there are multiple kinds of interneurons that are important in fine timing that could create the plasticity for the fear, the fear pathways and the safety pathways. So
yeah, that's
where my
exactly right. It's great points. Yeah, there's data on the VIP interneurons.
That's where I was going.
VIP, CCK. There's a number of different markers, and it kind of depends on which model system and when you're looking at it. So I don't think there's a grand unified theory yet, but I think there's a burgeoning literature on dynamic intergenerational regulation.
Thank you.
Thank you.
So next up is Genoveva.
Thank you, Dr. Kessler, for the wonderful talk. You know now that with COVID-19, there is a lot of PTSD and anxiety. And do you think these mechanisms that you described in the treatments would be applicable to anxiety after and PTSD after COVID-19?
Terrific question, very timely. Thank you so much for asking that. Yeah, we don't have any reason to think that anxiety, particularly trauma-related anxiety in one situation, be that a mass trauma, be that 9/11, be that war zone trauma, be that inner city, gun violence trauma, we think, and military trauma, the evidence shows that they're more similar than they are distinct in most cases. And certainly as a clinician, I think I would treat anxiety that was COVID related very similar to other anxieties or phobias. It's partly depending on what the specific symptoms are. But if, COVID related anxiety led to avoidance and sort of perhaps, inappropriate levels of fear about going outside or being with others or other things, I would treat it like other kinds of trauma. And I think a combination of psychoeducation, skills support, and trauma-focused therapies are the tools we have that seem to be quite good in those cases. Thank you.
Thank you.
So in the chat, we have a question from Bart, who says, do we know the functionality of the connection between the central amygdala and the substantia nigra?
That's a great question. I think somebody might know. I don't think I know the answer to that. We know more about central to VTA, again, from the dopaminergic systems. The VTA is more the limbic processing of dopamine, or the more ventral limbic processing of dopamine in amygdala, whereas the substantia nigra is more the dorsal striatum and more motor. So I know less about the amygdala to the substantia nigra role. In terms of the ventral VTA, there's definitely a dopaminergic role, and it goes in both directions. The amygdala can drive dopamine signaling, and dopamine signaling very much is involved both in fear conditioning and extinction. But we still we meaning the broader group field hasn't quite defined when is dopamine signaling enhancing fear conditioning versus when is it enhancing extinction, because it seems to at some level be related. I'm not sure if Marcus is involved, but there's a number of studies in Europe looking at dopaminergic enhancement of extinction using L-DOPA. And Mike Fanzelo and Michelle Krasky in UCLA have shown that reward learning in the aftermath of, at the end of exposure-based therapy that's thought to increase dopamine actually enhances extinction. So there's a lot of exciting new stuff coming out, but not a definitive story yet.
Thank you. So in the chat, we have a question from Stan Golden. fMRI studies of behavioral connectivity abnormalities in Alzheimer's and Parkinson's disease have provided useful insight at the human clinical level. Are you aware of any comparable studies in PTSD or anxiety disorders, such as changes in default, salience, and/or executive control network connectivity?
Sure, great question. So a couple different types of stories that sort of speak to this. We know, again, even at rest, so I talked a little bit about functional connectivity between prefrontal cortex and amygdala that support the basic idea that healthy emotional responding involves top-down regulation of prefrontal to amygdala, whereas overly threat responding and hyperarousal seem to have decreased medial prefrontal or subgenual amygdala connectivity and increased anterior cingulate and insula and amygdala connectivity. So that we see both at with threat cues like passive viewing of fearful faces, but you also see some resting state connectivity differences in those as well. Generally, you could probably make this argument for Alzheimer's and Parkinson's as well, but certainly in psychiatric diseases, even when we've got pretty robust group level data that are talking about some of the functional connectivity between reasons, none of that is robust enough for individual precision at the individual level. So none of these things are strong enough to be used in the clinic to say, okay, yes, because of this fMRI task, I can say that you as an individual have PTSD. But you could argue that even with tau and PET imaging and Alzheimer's, we're not quite there either. So I think it's been interesting contributions of that connectivity field that have definitely pointed to the circuit involvement that is also supported by the animal studies. Thank you.
Thank you. So next up, Charles has his hand up.
Hey, thank you. Very, very interesting. I was interested in your comments about the D-cycloserine, and you mentioned as I understood, the challenge of administering a drug intervention that has to be timed relative to a behavioral intervention. Your drug is only effective if the patient is in the right sort of cognitive state. I wonder if you could talk a little bit about the challenge of exploring that area. I imagine it must be quite hard to run a clinical trial with that sort of tool manipulation.
And therefore we're not. but definitely raise the, so I think the challenge is twofold. One, either we go broadly with plasticity related agents and we find a way behaviorally to really drive the memory towards, so I guess there's three options. One is if we knew how behaviorally to drive and preferential extinction of a fear memory instead of reconsolidation of a fear memory, then I would argue that you could use a BDNF plasticity agent or an NMDA plasticity agent and it would work. The problem is we don't really know how to behaviorally drive that. Another option is some of these drugs, because they work during the consolidation window of extinction, is you could wait and do the behavior and then see what the outcome is. If you have if you have an improvement of a certain amount during that exposure therapy, then I can be reasonably certain that you had a good extinction process. And now I can give you the drug if it can act within a few hours during extinction consolidation. And that's an idea that hasn't really been well tested out in humans yet. It's an interesting idea. The third idea that kind of I alluded to in talking about the cell type specific approaches is how do we go from plasticity broadly, which has the risk of enhancing multiple circuits that you don't mean to activating specifically circuits that only enhance extinction or inhibition of fear. So, one of our dreams would be, could you take, say, the TAC2 antagonist that we think blocks fear consolidation, or the beta-lactotensin, which maybe specifically activates the fear off cells? Could you combine those cell type specific circuits with a plasticity enhancer, and then pharmacologically kind of drive the circuit that you're interested in? So again, all of that's far away from human studies, but it's some of the interesting ways people are thinking about it.
Thank you.
Thank you. So a couple more questions in the chat. So a question from Frankie. You mentioned VTA connectivity and Mike Franzlaw's work on reward-based learning following extinction. Given the neural circuitry, BLA to nucleus accumbens, can you share your thoughts on how clinicians might combine therapeutic behavioral approaches to enhance extinction? For example, ERP and PE are unpleasant. Based on amygdala salience network connectivity, how would you link third wave approaches such as ACT and DBT might motivate engagement with ERP and PE? A lot
Awesome.
of acronyms.
Thank you, Frankie. You clearly know your psychological acronyms. Great work. Happy to talk off of this because I'm sure we could discuss more. But for the rest, the idea is there's a lot of different approaches to psychotherapy and trauma-related disorders, and ACT, acceptance commitment therapy, and DBT is more of an emotional-based learning. So how can you combine these? I do think Michelle Krasky and Mike's work would suggest that if you can do something, if we just kind of simplify it to what is it we're doing with our patients. Our patients don't want to talk about the trauma. It's unpleasant. It's a bad reminder. They're not sure it's going to work. They're afraid of it. Could you, I think some of what Michelle's saying is, can you combine that with kind of a positive reinforcer at the end? If we get through this, we get to talk about something pleasant, or you can have a pleasant mindfulness exercise, or we'll talk about very strong, positive social memory from the past. By doing that, it may both help restructure that memory network to take something negative to associate it with positive, but also may biologically increase dopamine at that time point when we think increasing dopamine might specifically help engage the extinction process. So that'd be one way I might.
Thanks. So another question on the acceptance and commitment therapy from Stephanie. I'm curious about modern uses of acceptance-based approaches, example ACT, in exposure therapy, especially in those where traditional exposure therapy is ineffective. Is there any evidence to suggest these approaches are operating through similar or distinct circuitry, or how to identify who might benefit most from such an approach?
Yeah, it's perfectly the question of where the field is. Can we take the tools we have now and find better biomarkers and imaging biomarkers to say who's going to benefit from what? The answer, I don't think, is that we can't yet. Studies are ongoing to do that. Other things we haven't talked about in humans are things like shame and guilt. Dissociation is a big deal that we see a lot. We know that dissociation gets in the way of trauma exposure, and this thing called EMDR, eye movement desensitization reprocessing, I think is broadly just a grounding technique that helps decrease dissociation, and that through decreasing dissociation, you also have better benefit from the exposure therapy. And it may be that shame and guilt act sort of parallel to that. When you have great self-hate and guilt, that you can't even let yourself go there because you're going so much down another path. And so it may be that that's some of the reasons why acceptance commitment work better. So clinically, when I see somebody who's very shame and guilt ridden, that's where I think the acceptance, the mindfulness, the commitment stuff can be particularly helpful beyond or in addition to the exposure, because they've got so much of that going on. It's hard for them just to sit with the trauma itself. Again, happy to talk more about that offline in a clinical way.
This is great to see a level of engagement and questions here. So let's see, we have another question from Genoveva. Since the identified cell types in the amygdala are in mice and animal models, is there evidence from humans that the same cell types will be involved?
Ooh, great question. You teed up our R01 and context integrants. So what's great about the single cell nucleus approaches that can now be done 10x and DropSeq and others is that you can really do this across species. So we're starting to see shared molecular architecture in some human amygdala samples and in mice. And then separately, in addition to the large scale genomics, we're involved and others are doing post-mortem. There's really with new molecular tools, sort of a reactivation of post-mortem work in PTSD. So we're doing a number of studies with several hundred PTSD brains versus depression brains versus controls, looking at proteomics, RNA-seq, epigenetic changes, et cetera, but can also start doing this at a single cell level. And what the real hope is that no longer will we have to be worried so much about whether our mouse model is truly a face valid model of threat or fear of PTSD. We can really just say, can I use the mouse microcircuitry of the amygdala to model the human microcircuitry of the amygdala? And then as I'm thinking about molecular targets for future interventions, I can do discovery. I can do discovery at the human large scale genetics level. I can do it at the human postmortem level. I can go to the mouse to really understand mechanisms and then prioritize the targets based on which ones we know are conserved in the right cell types. So while a lot of work is to be done, that's kind of how the field is thinking. Very exciting.
It's really exciting to hear about. I'm looking back, there are a couple of questions that people had submitted beforehand that I thought might be worth mentioning. You might have touched on it a little, but this question interested me. Is the formation or strength of fear memories influenced by diurnal variations or sleep?
Yes, we think so, but we don't know much about it. If you impair sleep after the first night of memory, you seem to get some level of decreased fear consolidation, but if you impair it before extinction, you also decrease. So part of the issue is we know hippocampal consolidation is critical for all sorts of things, and REM-based consolidation is critical. So to the extent there which is a contextual component of the fear memory, that can be disrupted by sleep deprivation. But similarly, hippocampal function is critical for extinction consolidation. So it's kind of tricky because it depends on which phase of the memory one is being involved. But so the simple answer is yes. It's important. The mechanisms by which it's important, we still don't well understand.
Thank you. So any final questions?
Well, it's been really fun. Thanks everybody for your engagement. Please feel free. I put my email, kressler@partners or kressler@mclean.harvard.edu. Happy to have offline discussions. Thanks for all your interest, and I look forward to meeting some of you in the future. Thanks so much, Parazad, for organizing.
Thank you so much for the fabulous talk, and thank you for everyone for your great questions. And yeah, thank you very much.
Have a great day. Thanks.
Bye.